All right. So this is chapter 8. This is the first part of looking at DNA. And this is going to look at the major structure and then also replication of DNA. So the DNA contains all of our genes in our body. These genes code for the creation of proteins, and they tell us which genes, other genes to turn on or to turn off. They regulate everything on our body. It's the blueprint or instructions that make you you. We have all of these characteristics that you inherit from your parents, and these are within these genes, these stretches within this DNA, coding for these different things. Okay. And so DNA is deoxyribonucleic acid. And we're going to go through and look at what each part of that word actually means and how that refers to the structure of DNA. So when we look at DNA, we all have this familiarity with this double helix structure, often referred to as a twisted ladder. It was first presented by James Watson and Francis Crick in Nature in 1953. They really were the ones that came up with this structure of this double helix. Now, Rosalind Franklin, however, was the one that really jump-started their ability to figure out this structure. She took an image through X-ray crystallography and saw these X patterns, giving them the idea that it must be a double helix within this DNA. She, however, rarely gets the credit that she deserves in the discovery of this structure. Here we see James Watson and Francis Crick building a model of what they think DNA looked like. And we can see these nucleotides, and we've got these sugar groups here. Okay. Again, Rosalind Franklin used the X-ray crystallography. Watson and Crick used model building based on what we knew was in DNA to figure out this structure. So we got the double helix model, and no new information has gone against this model for DNA. Okay, and here's her image that she took using X-ray crystallography of DNA, and we get this very distinct X, giving them the idea that the DNA crossed itself, but just in one spot. There were times where they thought it might be a triple helix, but then we would see a third strand coming in. Other times they thought maybe it would be a single helix where we wouldn't see this crossing occur. Chargaff's rule, this looks at the composition of DNA's nitrogenous bases. And what we see in this rule is that the A's, adenine, and T's, thymines, are always equal to one another. And the G's, guanines, and C's, cytosines, are always equal to one another. Okay. We were able to figure this out through various experiments before they knew it was a double helix. So they didn't even know that these things bonded together within DNA before these rules came about. Okay, and we'll see why these things are equal all the time. So these are the three parts of a nucleotide. And a nucleotide is the monomer or subunit of nucleic acids, so DNA, RNA, and so on. So nucleic acids are made up of nucleotides, and each nucleotide has three parts. There's a phosphate group shown here in the yellow. We've got a 5-carbon sugar, this dark blue. Now this is dependent on the nucleic acid. In DNA we have deoxyribose. In RNA it's just a ribose sugar. It's just a slight difference between those sugars. And then we also have 4 possible nitrogenous bases. In DNA, we've got thymine, cytosine, adenine, and guanine. Okay, so the 3 parts, phosphate group, ribose or deoxyribose sugar, and nitrogenous base. Okay, and that creates that nucleotide. Those nucleotides get bonded together to form that DNA. Okay. So there are 2 types two categorizations of nucleotides. You don't need to know these, but I will point them out because we'll talk about why pairing works out the way that it does. We've got our purines, thymine, and cytosine, and our pyrimidines, adenine and guanine. Okay. So we've got a phosphate sugar backbone. That phosphate group in the case of DNA, deoxyribose sugar, will bond together, forming the outside of this double helix. Okay, so that's our backbone. It keeps this DNA together, each strand anyway. Then we also see that there are two strands. This is a double-stranded structure, which is why it's a double helix. And then those nitrogenous bases are bonded together, kind of forming the rungs of this twisted ladder. And they are formed through hydrogen bonds. Okay, so again, another time that we see hydrogen bonds are really important. Okay, so we see this pairing shown here, and you want to know these pairing rules. Adenine is always going to pair to thymine, cytosine always to guanine. So if we look at this DNA shown here, we've got a thymine paired to an adenine, cytosine to guanine, guanine to cytosine, cytosine to guanine, adenine to thymine, and so on. These pairing rules are always true. One side of the DNA is always complementary to the other, following these pairing rules. Okay? So the reason that this happens is if we put two purines together, they would take up too much space. They extend beyond that backbone. We put two pyrimidines together, they're too far apart. They're not going to bond to the backbone and to each other. But you get a purine and a pyrimidine, okay? And this is where your hydrogen bonds form, and everything's going to be nice. So this is why we see this A to T and C to G. Okay? So all of this information, our genes coding for proteins and all of these other various things, is within our DNA. And it all has to do with the order of those nucleotides. Okay, our A, A, G, C, T, C, G, whatever order those nitrogenous bases are in, those are the instructions. Okay, and so we've got enzymes that can read those instructions and build the proteins that we need, which we'll talk about here in part 2 of DNA. So DNA replicates itself. Okay, it makes copies. So why does it do this? And the reason is because our cells replicate. We make new cells. Each one of those new cells has to have an exact copy of that DNA if it's going to function the way that it needs to. So we replicate that DNA before we replicate those cells so that each of them can have an exact copy of that DNA. And so here we're going to look at a basic illustration of how DNA copies itself. So we've got our parent molecule. Here it's just showing 5 nitrogenous bases for simplicity. Those strands will separate. There's an enzyme that does this. It's called DNA helicase. We'll see it again on a slide further down. But we separate these strands. Then DNA polymerase, another enzyme, comes in and it starts copying the complementary bases. So where we have an A, it's going to bring in a T. Where we have a C, it's going to bring in a G. Where there's a T, it's going to bring in an A, and so on. And then on the other strand of that parent molecule, it's going to do the exact same thing, bringing in complementary bases. And so as we do this, we create 2 new strands that are identical to the original strand of DNA. And so we call this semi-conservative replication because half of that original parent is conserved in each of our new strands. Okay. So looking at these steps, we have to unwind this DNA. We have to open that up, and this area where it separates is a replication fork. This is done by DNA helicase, an enzyme.